So it's easy for me to talk about the subject because many have introduced already high C and basic features that we're seeing in high C. And I will just remind you what we covered in our tutorial before this conference. Essentially, there are two types of patterns that are distinctly visible in high C. Those off-diagonal checkerboards that Aris also just spoke about, compartments, and near-diagonal patterns. And what we demonstrate over the years is that these checkerboard patterns that refer to as compartmentalization, separation of euchromatin from heterochromatin in space, are largely driven by attraction between heterochromatic regions and not by the attachment to the nuclear envelope, though they are attached to the nuclear lamina. And as far as the loop extrusion, again, what we, I'll just go straight to the model. So what we proposed is that that loop extruding factors, at that time hypothetical, can land on DNA and form progressive larger loops through extrusion and then stop at CTCF sites. And so when we simulated this process, what we saw indeed is that there are maps that remarkably resemble high C maps. At some point, we were even discouraged by having these strange stripes and dots that were not really evident in the data at that time. And then ERISIS 2014, high-resolution data came out that remarkably saw these individual dots and stripes and other interesting features. And we concluded that loop extrusion with boundaries can indeed reproduce these near-diagonal features. And that kind of formed our, I would say, kind of basic loop extrusion model. And over the years, we kind of learned many new things. Just to kind of highlight, so evidence of loop extrusion also came from a number of kind of interesting directions. For example, we were interested in the role of loop extrusion as a mechanism for mitotic compaction of chromosomes, and that's the work of Anton Goloboroidko, who is a PI at IMBA in Vienna. So when he was in my group, he simulated this loop extrusion of excessive number of extruders that are not being stopped by CTCFs. And what we found is that this high level of extrusion leads to essentially kind of extended and compacted structures, kind of sausage-like structures, which certainly resemble my kind of prophase chromosomes, can also segregate chromatids. But at the same time, it can also reproduce the phenotype that Jan Michael Peters' group have seen earlier that they called vermicelli chromosomes. So what happens if you overload cells with cohesin or overload chromatin with cohesin by removing the protein called WOPL that unloads cohesin from chromosomes. So formation of these dense structures was very hard to reconcile with then believed role of cohesin in just cohesin to sister chromatids. But this formation of these dense structures was perfectly consistent with loop extrusion model. But again, decisive experiment for loop extrusion was certainly Case's single molecule experiments, famous Ganji et al. Paper, where they've seen, and you've seen this two talks back, this beautiful formation of extruded loops in vitro. So that's where we were, and then in 2019, Jan Michael Peters' group also demonstrated, using similar single molecule experiments, that cohesin can extrude loops. Now the question is, how does loop extrusion operate on busy DNA? So how do SMC motors interact with bystanders, other proteins on DNA, and so how SMC motors can interact with each other? And in this sense, kind of my talk will be very much rhyming with Case's presentation, at least this part of my talk, where we're trying to ask similar questions, but not from single molecule perspective, but more from observations in living cells, and then trying to see where we do or do not agree with single molecules. For example, an interesting study that came out last year, again in collaboration with Jan Michael Peters and Kiko Itachibana, was a study of proteins, replicative helicases, MCM proteins. So these are parts of the replicative machinery that are present on DNA in G1. And it's not entirely known what they're doing there because it's way before replication. Nevertheless, what Kiko Itachibana's lab has demonstrated is that removal of MCM, removal of these helicases, strengthen all these features in high C. So this is a wild type versus removal of MCM in the maternal zygote. This is, again, the similar effect in the paternal zygote. As a note, this is done by single nucleus high C because you cannot certainly accumulate enough of zygotic cells. So this is done after fertilization in maternal and paternal zygotes. So the question is, how can these features get stronger? Somehow this effect of stopping cohesin becomes stronger. We tried many models, and the model that we converged on that reproduced this data was an idea that MCMs essentially work as very weak and randomly distributed barriers. So beyond CTCFs that are well positioned and establish very strong and patterns visible in high C, there might be many that are random. As a result, they're different in different cells and at different moments in time. So they leave no obvious footprint on high C map. Nevertheless, they slow propagation of cohesin. And when they're removed, cohesin can get to CTCFs earlier and get stronger features. And so that's what we've seen also in our simulations, that removal of random barriers strengthens these features. So the lesson learned, there can be random barriers and other proteins that are not serving specifically to stop cohesin can nevertheless be barriers. As a note, actually, related to cases talk, MCMs form helices that wrap DNA, that wrap around DNA. So it would be interesting to see whether this class of proteins are better stoppers than others. Okay, looks like so. Another paper that we published with Jan Michael Peters' lab was an exploration of what's the effect of transcription on loop extrusion. Again, in the context of removing CTCF as the strongest barrier and increasing the abundance of chromatin-associated cohesin by the Wapol knockout, we examined high C structures next to the genes. And that's how these patterns look like. So what do we see immediately from that? We see that the gene is a nice insulating point. You see this kind of reduction of contacts between regions upstream and downstream from a gene. This effect gets stronger for more highly expressed genes and for longer genes. And moreover, there are very specific patterns that are formed around the gene if you orient these maps in the direction of transcription. And that made us suggest a model where polymerases, with their, presumably the whole complex of polymerase with a large RNA emanating from it, can pause transcription or can pause loop extrusion, but in a very peculiar fashion. Because cohesins move so much faster than polymerases, as we know again from single molecule experiments, they would stop if they go in the same direction as polymerases. So if they go in the same direction as polymerases, so here polymerases go in this direction, so cohesin would be catching up with a polymerase really fast, and then moving with the polymerase at its slower speed along the body of the gene until it bypasses the polymerase. In the head-on collision, cohesins would meet polymerase in the middle of the gene, but again, from single molecule experiments, we know that cohesins are relatively weak motors and polymerases are relatively powerful motors. So polymerases would outpower the cohesin and would be pushing it back, at least this part of the cohesin, and until again cohesin bypasses the polymerase. So we decided to test this model to see whether it can reproduce, under what conditions it can reproduce this data. It's relatively difficult to reproduce this average high C maps. The best models could achieve again agreement in the overall insulation and generate the stripes that we're seeing. Some of the features that we see are really faint in the models, but at least we believe that we're on the right track to understanding how transcription can interfere with polymerases. And the lessons we learned is that Pol2 can be pushing cohesin, but for merely less than a minute. Because if it were pushing it too strong, again, we wouldn't see this pattern. And cohesins do not loaded promoters. That's another interesting result from this study. We predicted this from the model that it works only if cohesin is loaded uniformly. And indeed, when confirmed this by kind of new generation of ChIP-seq experiments on tagged NIPBL proteins. So if you're interested in details, I encourage you to look at this manuscript, at this paper. Another interesting lesson we learned, again, so we now know that random certain class of proteins can serve as extrusion boundaries. actively transcribed genes can serve as extrusion boundaries in an interesting fashion. And then that was a collaboration with Zindan Wong's lab, where we looked at structures that emerge in bacteria when extrusion, again in bacteria, extrusion starts at specific positions close to the origin. And in Bacillus, you can actually move this site of loading of an extrusion factor to a new position. If you move it here, you will see this hairpin emerging in a new position, representing kind of extrusion from one place, and it kind of propagates along from this point on. Kind of, I don't have a cable, so imagine I'm operating with an imaginary one, and I wouldn't risk touching this. Now, what's interesting is that if you move this site to a new position, you see hairpin emerging from another position in this bacterial chromosome. But if you have two of them, then instead of just two hairpins, you see much more complicated star-like pattern that emerges. And we tried many models to reproduce this, and the only one that worked was the model that was basically the mechanism that was described by this paper by Case Decker and Eugene Kim, where they observed that two condensing molecules can bypass each other while they're extruding. And we say, what if these bacterial condensers can also bypass each other? And indeed, if they bypass each other rather than block or trigger the unloading, you can get this star-like shaped pattern. And that was a strong indication that in vivo, what we're seeing is this phenomenon of condensing bypassing. Whether this is true in mammals, whether this is true for cohesins, remains to be seen, because it's hard to reproduce similar phenomenon, again, of target loading in mammalian systems. So in another study, we decided to revisit the original notion of dynamic loops. So what we proposed in our kind of original formulation of loop extrusion was that we estimated loop sizes. We also said that loops should be very dynamic, and about 50 to 70% of DNA should be in the loops during interphase, otherwise interphase chromosomes would become compacted. We also estimated the fraction of time a particular large loop, we say 400 kb, would be present, and say these dots that we see on high-C maps are not loops. These are places that can come together and stay together for short periods of time. We estimate about 7% for this 400 kb region. In collaboration with Anders Hansen at MIT, we examined live cell dynamics of two regions that are labeled, have CTCFs on their sides. They're separated by 505 kb. And you can track their dynamics in live cells, and so then analyze this data. And so the analysis was done by Simon Gross-Holtz, who just graduated from MIT. So this data basically, in the experiments, you can track two loci. They move relative to each other. We're looking only at their relative distance, or at their distance, strictly speaking, and as a function of time. So we get this trajectory. And from this trajectory, we can infer the looped state. And it's not inferred by just proximity, by imposing some cutoff. It's inferred through kind of by examining relative fluctuations of these two loci. And again, I wouldn't go into details of the physical model there, but that allows us to say when they actually move together and fluctuate as if they're bound by a bond. And that gave estimates of the looped fraction, which turned out to be indeed about 6 to 8% in the wild type. The looped fraction is high if you increase the residence time of cohesion on DNA, and the loop fraction drops to roughly our error of measurement of about 1 or 2% if you remove CTCF or if you remove cohesion. So these loops are incredibly dynamic. So the lessons are that also we estimated from this data that about 57 to 61% of DNA is in the loops. That this loop separated, again, this is a relatively large loop, half a megabase loop, is present only 7% of time, and is present there for only 10 to 30 minutes. Basically, once in a cell cycle, in a 24-hour cell cycle, these two things come together, stay together for about 10 to 30 minutes, and then dismantle. Most of the time, it's not a loop. And that CTCF, another interesting thing, so CTCF was modeled here, answering question that Case asked Aris. So we modeled CTCF as something with about 50% permeability. So it catches about half of the molecules that come to CTCF. Here there was an array of two CTCFs on one side and another CTCF on the other side. What's also interesting that to reproduce this data, we had to also impose a rule that CTCF extends the life of cohesion that is bound there. And this also agrees with the structures solved by Ben Roland's group. Okay, so I will make an announcement of a talk that my postdoc, Alex Golitsina, is giving on June 8th. So what Alex will be talking about, new structures that we started seeing in high C under certain conditions. We call them fountains, so because they look like fountains. As a mechanism for their formation, we propose a number of possibilities, including the possibility that there are random barriers, because if you just had cohesion loading over here, it would be a hairpin like in bacteria. Nevertheless, these fountains tend to broaden. And the question is what can make them broaden? And our hypothesis is that there are some random boundaries that can broaden it. These boundaries can be other proteins that we've seen again can be present on chromatin, or it can be other cohesions that actually load in not at this targeted site. Another possibility, and again very much in agreement with what Case just spoke about, is that cohesion by itself is a bit of a random kind of drunk extruder. So it extrudes a little bit on one side, then extrudes a little bit on the other side and keeps doing this. So essentially doing some sort of a random walk here instead of a straight line that bacterial condensers can form. It's also possible that it switches direction, as we were again also hypothesizing, and it's nice to see that there are evidence of the switching behavior now, emerging now. But I will let Alex tell you this exciting story and where we see these fountains forming. So these are several lessons that we learned in recent years about interference of extrusion and other process on chromosome, transcription, other factors, compartmentalization included, and so on. So there are many new rules of extrusion that we're learning by analyzing high C and microscopy data. In the last few minutes, I will tell you about our work that kind of more extending the talk of Andy Spekowitz than other talks, because we were interested in the question of maintenance of epigenetic memory, but generally interested in the question of we have good now understanding of near-diagonal structures in high C. Do we understand these large checkerboards that we're seeing, compartmentalization? What might be the function? And the hypothesis is that compartmentalization can help maintain epigenetic memory. What do we mean by epigenetic memory? So imagine a situation that something came and established specific histone marks on DNA, and then you track this through many cell divisions. Does this pattern, without being maintained by external factors, can this pattern be maintained? Like the way you save a file on a hard drive and it stays there, maybe accumulating some mistakes in this case. So can this memory be self-sustained for many cell divisions? Why is this challenging? It's challenging because histone marks are being constantly diluted by replication. When you replicate, even if you retain all the labeled histones, which presumably you retain some fraction of them, then even in this case you get 50% dilution of all the histone signal. There are enzymes that can recognize a marked region and impose a mark to a nearby histone. So textbook gives you an answer saying there are read-the-right enzymes. Is this enough? And so again, we model this, again, in the same way as many others, as an enzyme that recognizes a mark and imposes a mark on a nearby histone. Can this really create a memory that is stable? To answer this question, we examined a class of models, again, considered by several groups, where you have the spreading of the mark. The mark can be spontaneously lost or lost during replication, depending on the flavor of the model you consider. So there is a spreading rate S, there is a loss rate L, and these marked regions also tend to be attracted to each other, because we earlier demonstrated that heterochromatic regions tend to attract each other. So as an additional parameter, we'll have the strengths of attraction between marked regions, and I will refer to them as B regions. So two essentially parameters, two dimensionless parameters. That's what controls behavior of this model. And again, many again, Andy Spekowitz, David Morinduzzo, Daniel Jost, Fabian Erdel, and many others have considered this class of models and have seen problems retaining the memory for many generations. And so we decided to revisit what's going on and can we find conditions that would enable long-term memory. And we considered two situations. One is with 1D spreading of the marks, and another was the spatial spreading of the marks. In the case of, and again, the setup will be such that I would impose the marks let the chromosome fold, and then stabilize the chromosome during interphase, and then let the marks spread. And then during metaphase, I will do the opposite. I will keep the marks and refold the chromosome completely, and it will go into the next cell cycle. So if we do this when we allow 1D spreading only, there is no stability in the system. So what I'm showing you here, time in cell generation, and this is the pattern of marks that you establish. If S over L parameter is lower than a certain cutoff, again, known in physics and mathematics, then there is a loss of the marks. If it's greater than this magic number, 1.6 something, then everything gets marked. So there is no stability in the system, and people have seen this. Now, with the 3D marks, can it work? For one generation, indeed, if the polymer does not move, so it folds and then it stays, then you have perfect preservation of the mark. Then it stays there. If you, again, not for any values of parameters, but for a range of S over L parameters, it's stable. So we have perfect memory for one generation. What's going to happen if I were to iterate? So again, this is just to show that in one generation, you can, for example, erase or introduce random marks, and then the original pattern comes back. So it's a very stable system. But if you start iterating, then things break down. After about 5 or 10 cell divisions, the memory is completely lost. So it doesn't hold the memory. And again, it's similar to what Andy has observed in his PNAS paper, that there is memory for about 5 generations, but beyond that, it doesn't really, it starts to deteriorate. And we examine the space of parameters S over L and alpha, trying to find maybe there is a regime weight stable. And yes, you can kind of find the regime weight stable, but it's like balancing a pencil on its tip. So there is a narrow regime where the memory can stay for maybe tens of generations, but it generally has a tendency to either shrink or expand, these domains. So they're still not stable in a sense of kind of hundreds of generations. So then we realized there is one thing that is missing in these models. And this one thing is the concentration of the enzyme. We assumed that the spreading can happen anywhere in the nucleus, independent of where else it's happening at the same time. And we say, well, that cannot really be the case because you have a finite pool of enzymes. So let's introduce a finite pool of enzymes that lead to the spreading of the mark. And when we've done that, what we see is a very different picture. Then an imposed domain can stay essentially in the same place, wiggling and jiggling, but still staying in the same place for hundreds of cell generations. And when we swap the parameter space, we see that in a broad range of parameters, you see very, very, very stable memory. What's required for this is sufficiently large alpha. And our exploration of the one generation system argues that the alpha should be sufficiently high such that the density of heterochromatin is high. Then the marks stay within heterochromatin and they do not spread everywhere, and you need a limited enzyme. So there are interesting phenomena in the system. It can maintain several domains. It can also see what Andy was referring to today, like short moments when some marked regions appear and disappear, and we didn't quantify the size of that, but it's a really, really clever idea of what you've done. You measured sizes of the spontaneously emerging things. And that there is some sort of kind of dreaming of the system. So there are things that appear and disappear, or error correction, or kind of automatic error correction. They lost after a few cell divisions. And again, this memory is not perfect either. After many hundreds of cell divisions, you start losing the memory. And again, so that's presumably that's a pessimistic thing, there is nothing permanent in life. And that's another phenomenon where you cannot really maintain this memory forever. There are other interesting phenomena such as positional variegation, position effect variegation in the fly of Drosophila. Without going into details, I'll just say that this gives you kind of random but stable heritable patterns. I summarized what we learned from that. We need 3D spreading, we need dense B compartments, and we need limited enzyme. And that's what can lead to stable memory. And as far as extrusion, there are lots of new rules that we learned since 2015. And these are the members of the group. And the group is getting smaller. And there are more alumni. And Henrik, Emily, and Sasha are here. I'm very grateful to the organizers for inviting me and for supporting their travel. And feel free to ask questions. Thank you.
So so if you look at one domain, if you look at it over many generations, I guess it diffuses. Did you look at what type of diffusion it has?
Excellent question. We looked at this diffusion, and we don't understand what's going on, because you cannot really define it as a boundary. You can define the total number of marks. So it seems to be stable. So at least this merger we could not explain by diffusion, and the lifetime we could not explain by diffusion. Because if it were diffusive, it would scale like a square root of size, and it scales linear with size. So there is also another phenomenon of competition between domains. And when one gets bigger than the other, it starts kind of sucking monomers from the labels from the other. So there is a diffusion, but there is also a drift effect, presumably. Yeah.
Hey, Leonid. Thank you. That's really interesting to think about. I was wondering if, maybe I didn't understand this correctly, but did you take into account the length of the cell cycle and what would happen if you know, in fast cycling cells?
Yes, that's a great question. So we did look at this. In fact, when there is replicative dilution, the number of marks that you can restore through the spreading depends on the duration of the length of the cell cycle. And indeed, if the cell cycle is too short, that would lead to unstable memory, or you would have to reduce your spreading rate. If it's short, you would have to increase your spreading rate. So you need, essentially, the prediction is that depending on the duration of the cell cycle, you need to adjust the activity of these enzymes. Or the quantity, exactly. Yes.